Our last topic for this unit is to talk about these things called viruses. And by the time we're done, we should be able to figure out, are they really living? And if they're not living, then obviously we will not classify them. So we're going to go through some information about them, and by the end you should be able to decide that. So here what we have is a picture of 3 typical viruses. We have one here, it's the tobacco virus, so obviously that would infect plants. We have something called a bacteriophage, which would be a virus that infects bacteria. And we also have an HIV virus, which we know HIV only infects animals. Now, what I hope you notice here about the viruses is that they have a couple things in common. They all have some sort of genetic material inside of them. So here we have RNA. RNA is a lot like DNA. We'll talk more about that later. Inside the tobacco virus, we have DNA, so there's our RNA. We have DNA inside of what's called a bacteriophage, and inside of HIV, we also have RNA, again, another type of genetic material. Viruses are also enclosed around some sort of protein coat. So there's some sort of protein that will surround, not a cell wall, not a cell membrane, but some sort of protein that will surround that genetic material. So you can see here again, these proteins. Now, some viruses, like animal cells, have these lipid envelopes that can be used to trick the host cell into letting the virus in. And the idea is, and we'll talk about this on the next slide, that viruses need a host in order to start to replicate. So hopefully by looking at the pictures and listening to this, you'll be able to fill in the information. And really the bottom line from this page is that viruses have this general structure. They're not cells, but they have genetic information surrounded by protein, and they're very specific about what they can infect. So we cannot get the tobacco virus because we are not plants. Now here on this slide, what we are looking at is the typical reproductive cycle of a virus. Now in this case, we're going to look at a bacteriophage, which is a virus that infects bacteria. There are 2 different cycles that it can go through. And letter A is pointing to what is called the lytic cycle. And the lytic cycle follows this 1 to 2 to 3 to 4 and then back to 1. And letter B is showing the lysogenic cycle, which kind of starts here at 1, it goes to 2, then jumps over here to 5, and then 6, and then 7. So let me talk a little bit about each one, and then as I talk about it, you can fill it in at the letters below. So here at step 1, what is happening is the virus, in this case the bacteriophage, is attaching itself to the host cell. The host cell is this right here, which is the bacteria, and it's going to inject its genetic material. That's what you see going on there. Now in step 2, what you see happening is the genetic material has now completely entered the host cell, and it's going to start to take over the host cell. And when it takes over the host cell, it's going to tell the host cell, stop doing what you're doing and start making more viruses. And that's what's happening here in step 3. The host cell is making more viruses instead of doing what it's supposed to normally be doing, which is functioning and making proteins and whatever else it has to do. Now, it will get to the point where there are so many viruses made that step 4 will happen. And in step 4, what's happening is all of the viruses that were made are now lysing or breaking out of the host cell. And what do they do next? They attach to another host cell, and the cycle keeps on occurring. This is called the lytic cycle because it's infectious. And so what that means is it's causing the host cell to be infected, and in this case, the host cell will end up dying. Now, lysogenic, little different. So we start with 1. The virus is going to inject material into the host cell. The genetic material here at step 2 is now completely in, but instead of taking over and moving to step 3, the genetic material will blend in with the host cell material and become what's called a prophage. So a prophage is when the viral DNA blends in with the host DNA and it's dormant, like a volcano. So it's almost like it's in there, but the host cell doesn't know it's there. So now when the host cell divides, which is what's happening here in step 6, it divides with the viral DNA inside of it. And this can happen forever and ever and stay dormant until one day, step 7, it comes out from dormancy. That's what's going on right here, and loops into the lytic cycle. And so a great example of this, now this is how it would work in bacteria, but let's use humans as an example. You may have heard of herpes. There's herpes simplex 1 and herpes simplex 2. Herpes simplex 1 infects, I always say, from the waist up. So really herpes simplex 1 is what gives you cold sores. And you may know some people who get cold sores all the time, and you may know some people who never get cold sores. Well, I'll tell you right now, you probably have that cold sore virus in your lip cells right now. It's just that right now, it's dormant. However, something can trigger it to come out of dormancy. So stress, hormone changes, food that you eat, you get sick. And when it comes out of dormancy, it goes into the lytic cycle, and that is when you end up getting the actual cold sore. So once in lytic, it's always in lytic until your body fights it off. But lysogenic can go from lysogenic to lytic. But lytic can't go into dormancy. Once it's lytic, it's lytic. Okay?